Alright, hello SBI4U. This is the second video in the playlist, and it's your first actual new content for the day. This one will have a quiz associated with it that's going to count as 1 hour of class time once you complete the quiz. Let's talk about the learning goals. We're going to jump right into it. Today we're talking about the importance of proton pumps in the mitochondria as part of the electron transport chain. We're going to talk specifically about NADH and FADH2. We'll talk about their structures and what they do and how they bring energy into the mitochondria. We'll talk about the pumps themselves, each of the 3 pumps we will cover in detail, and then the 2 electron shuttles, these very important molecules that are very useful to the mitochondria. And then finally, a little bit of application. We'll talk about animals that can uncouple the electron transport chain. And we'll talk about stuff like brown fat and a drug called DNP. So jumping over, we're going to take those away, put up the current topic, and keep in mind, and we'll jump over to this here, actually, this one. So first things first is every time you hear me say ETC, what I mean is electron transport chain. ETC. Yeah, it's just a shorthand. You'll see that a lot. And this is a diagram of it. I'm going to bring this back over to the right so you can see the diagram a little bit better. You see here, you see here the NADH, and it's crossing over. It's releasing its hydrogens. 1 hydrogen, and the electrons are coming through. So the red here is the electrons, which is why it's called the electron transport chain. It's called a chain because there's 5 different molecules that grab the electrons and push them along. So that's what's called a chain. It goes from this pump to this shuttle to this pump to this shuttle to this pump, and then finally goes on to oxygen, which is the final acceptor. These pumps sit across the membrane, and they pump hydrogen. This is why they're called proton pumps. So if you look down here, this section is called the importance of the proton pumps. So let's jump right into why these proton pumps are important, and I'm going to move over to the document camera view. There we go. And I'm going to move the camera. It might be a little bit loud, sorry guys. Ooh, that doesn't work. That works. Let me know if that's too loud, and I'll make it better on other videos. You know, always feedback. This is all new for me too. So here we have a diagram, a simple diagram of a mitochondria. So this entire black section is just a mitochondria. This whiteboard here might be considered the whole cell, maybe, or part of the cell. You know, maybe there's a nucleus somewhere, maybe there's some Golgi body somewhere. You know what? Let's actually draw a nucleus up in the corner somewhere, just to be like, hey, there's a nucleus up there, right? Nucleus. Right, there's a nucleus there. Maybe there's some ribosomes around. Right, this is like inside the cell. Please don't think that this is just a cell. This is actually the mitochondria. This black thing that I've drawn here is the mitochondria. And where we left off was talking about this gradient here, right? We've got a lot of H plus in the intermembrane space, right? In that intermembrane space between the inner and outer membrane, and not a lot of H plus in the matrix, which means that we've got a gradient between the intermembrane space and the matrix. We call it an electrochemical gradient, right? A proton gradient here. Meaning that these protons, because of the charge and because of the because of the concentration, they want to flow in. But we don't let them flow in, right? We've got the membrane in the way, unless they go through this red, these red little turbines. And those red turbines are ATP synthase, right? ATP synthase. Those are the red turbines, ATP synthase. But, you know, think about it. This section is called the importance of the proton pumps. So what happens if we just have this system? We're going to make a bunch of, we're going to convert a bunch of ADP into ATP, but then all of these will flow in and we'll be done, right? We won't have any more gradient. We won't have anything to push stuff through the turbine, and then we won't be able to convert any more ATP. So we need some way to recharge the proton gradient. And that's done using the pumps. Now the pumps are kind of all over, and so are the ATP synthases. They're all over this membrane. But just for the sake of you know, a clear diagram, I'm going to put the pumps over here, and I'm going to keep the turbines on this side. But yeah, these pumps are everywhere on this membrane, and the synthase is everywhere on this membrane. This process is happening constantly. So we have these pumps. Right, I'm gonna make them little blocks, and the pumps, their job is to take the H plus from inside and push it back out. Right, take this H plus from inside and push it back out. So basically, yeah, they recharge the gradient, keeping this high, keeping this low, and making sure that there's a driving force to make that turbine move. And when the turbine moves, we get ATP, and our cells use that to do all kinds of things. So so that's the pumps. These are the proton pumps. I think I lost a red, yeah, there we go, lost a red pen. These are the proton pumps. Right, that's their importance. They have to keep cycling it. And as mentioned before, the way that they do this is with electrons. Electrons actually provide the energy. So something has to give them electrons so that the electrons can move through, right? The electrons move through. It's actually 2 electrons at a time that move through. And something has to release those electrons. And that's where our energy carriers come in, right? What's going to bring up these electrons? Well, that's going to be the NADH and the FADH. So let's draw those. Let's add those. Right? The NADH and the FADH will provide the electrons. Right, so those electrons actually come off of NADH, and then they have to go somewhere. The electrons have to go somewhere at the end of this whole pump setting, this whole pump system. And if we jump back over to the diagram that's in your note, let me just jump back over to the diagram in the note. You see that the final acceptor here is O2, oxygen. All right, its main job is to accept those electrons and then leave. Because if it didn't, then the electrons would get backed up. Right? It's like a traffic jam on the highway. If nothing's accepting them here, then this thing can't take them from C, and then C can't take it from this one, and then this one can't take it from Q, and then Q can't take it from this, and then this can't take it from NADH. So if you don't have something to accept the electrons at the end to clear the electrons, the whole thing gets backed up and nothing can move. And then no protons go, and then you don't maintain your gradient. So that's the importance of oxygen here. We breathe in oxygen just to clear those electrons away from our electron transport chain. So jumping back to this guy, here it's the electrons are going to attach to oxygen. So the 2 electrons plus 2H plus half an O2 O2. You know what, that H plus should be in blue. Because I've been using H plus in blue too. H plus plus half an O2, and that becomes water. And the water is going to leave. So, you know, double duty here. Number one, oxygen will take away the electrons to allow more electrons to flow in, to allow more things to pump. And number two, it's actually clearing H plus from inside the mitochondria, from the matrix, which means that we're reducing this concentration even more, pushing up this gradient even more. So the pumps and the whole electron transport chain are very, very important, and their entire thing is maintaining the gradient, keeping the gradient going, making sure that this part of it can still happen, right? But they are two completely separate things, right? This can happen even if this is shut down for a little bit for some reason. You know, if you don't have enough oxygen, you can still run this for a little bit. You'll just build up H plus in here, and then later you'll have to use oxygen to pump it back out again, and stuff like that. So that's the importance of the proton pumps in the mitochondria. Let's jump back over here, and we're going to change over the current topic to NAD and FAD, right? NAD and FAD. NAD and FAD. Keep in mind these. So NAD and FAD are what we call the electron carriers. These electron carriers, right? They're electron carriers. Go through. They go through a similar cycle as ATP and ADP. So, you know, you're going to see them kind of flip back and forth. So what I've got here is, in my Google, here we go, is I've got pictures of them. So here we have ATP. We've seen this picture before with A and then the three phosphates, ATP. Let's look at NAD. And just as a reminder, when nature figures out a way to do something molecularly, it likes to keep a similar way to do things. So, hey, this transfers energy. NAD also transfers energy, and you'll see a similar kind of structure. Here's the A, here's the adenine, here's the ribose, and then there's two phosphates. So this is like basically ADP, but then instead of the third phosphate, there's another ribose, and then it's attached to this. And this is called nicotinamide. So NAD actually stands for nicotinamide adenine dinucleotide. So dinucleotide because there's two riboses and two things attached to the riboses. Nicotinamide because this is nicotinamide. This is the cycle that it goes through. Remember with ATP, it loses the phosphate and becomes ADP. Here it's not a phosphate that gets transferred, it's actually a hydrogen. So this one here is NAD+, it's a positive. You'll see here this hydrogen attaches or detaches, and it attaches up onto this carbon here. One interesting thing about this is this hydrogen that comes in is actually carrying 2 electrons. So the hydrogen is carrying 2 electrons. So on NAD, it only grabs 1 hydrogen and attaches 1 hydrogen to itself. But that hydrogen carries both electrons, which is neat. In order to do that, that hydrogen steals an electron from another hydrogen, and then that hydrogen is released out into the solution. So that's why we have NADH plus H plus. The H plus makes the solution slightly acidic. So this NAD cycle is actually kind of an acid-base type cycle. One thing to note, it kind of looks like 2 hydrogens go on here, but that's because this is the organic chemistry shorthand. If we look at this carbon, right, thinking back to Unit 1, it's got 1, 2, 3 bonds to other carbons, which means that there's a hidden little hydrogen there already. So one of these hydrogens was already there. The second hydrogen was the one that got added on. So a hydrogen with 2 electrons joins in, making both those electrons make the bond. So this thing is now carrying a hydrogen and 2 electrons. That's NAD. Now once again, we're gonna when nature figures something out, it likes to do the same thing. So let's talk about FAD. FAD looks a lot like NAD. Right? You've got your adenine, your ribose. So there's the A and there's the ribose. 2 phosphate groups. Excuse me. 2 phosphate groups, and then this funky like glycerol thing going on, and this guy. So FAD is very similar. It's still a dinucleotide, which is the AD. It's an adenine dinucleotide, but now it's flavin. Flavin is the molecule here. This is a flavin molecule. These molecules come from just vitamins that you eat, and it's B3 and B2 will make these. So it's called nicotinamide, but that doesn't come from nicotine. It comes from vitamin B3, which is where is it? Niacin. So yeah. So that's what they look like. And FAD, unlike NAD, FAD actually pulls single hydrogens. Now these are hydrogens with 1 electron. So remember here the hydrogen had 2 electrons. Here each hydrogen just has 1 electron, and they go on 2 different nitrogens. So you see here this nitrogen is full and this nitrogen is full. Remember, nitrogens make 3 bonds, so these nitrogens are good. What happens when the hydrogens go on is these double bonds kind of shuffle around, and then there's room for that extra hydrogen. So really this is just 1 hydrogen on each nitrogen. So FAD accepts two hydrogens. NAD only accepts 1. So let's do that as a keep in mind. Let's change our keep in mind to that. Keep in mind NAD accepts 1 H and 2 electrons. FAD accepts 2H and 2 electrons. Right, keep in mind. Right, which means that the hydrogen that gets accepted by NAD has that hydrogen gets accepted by NAD has to have both electrons with it, but the hydrogens that get accepted by FAD have 1 electron each. Which means that this molecule, because this bond is kind of like weird because the hydrogen came in with 2 electrons, it's like a higher energy bond. Those 2 electrons are like, they want to leave more than each of these electrons want to leave. So NAD is a higher energy molecule than FAD. NAD is higher energy. So that's what NAD and FAD look like, and why NAD is higher energy than FAD, because in NAD both of the extra electrons are stuck in 1 bond, and in FAD they're kind of spread around in this resonance structure. Okay, so that's that. Let's jump back to our learning goals. We've talked about the proton pumps. We've talked about NAD and FAD. I forgot to mention something about that. Let's talk about the charges. NAD actually starts positively charged. This nitrogen has a positive charge on it. It's making 4 bonds. And then when the hydrogen comes in, it comes in with a negative charge, so the charge neutralizes out. But in order to come in with a negative charge, that hydrogen pulled an electron off of a different hydrogen, giving it a positive charge. So that's why we get this charge balance here. But FAD accepts neutral hydrogen because the bonds just shuffle around. So FAD just becomes FADH2. There's no charges to think about there. There's no charges to deal with. So that's another reason that this one is higher energy than this one, because if you can do an electrical rearrange, you'll end up with a higher energy molecule. Okay, back to this. We've done NAD and FADH2. Let's talk about the 3 proton pumps. Let's get into the 3 proton pumps. our topic, the three proton pumps. And let's talk about our, keep in mind, the pumps use energy from NAD and FAD electrons, but not necessarily their H, the protons. Is that going to go off screen? That does go off screen. Okay. Let's try that. There we go. Not necessarily the protons. Huh. Oh, that's weird. Oh, I guess I should call it NADH and FADH too. Okay. Right, so the pumps use energy from NADH and FADH2 electrons. So the electrons are actually what's going to drive the energy of this reaction, but they don't necessarily use the protons from NADH and FADH2. This is what I mean by this. Let's take this away and we'll go back to this view. What I mean by this is, if I go here to the diagram of the whole process, is notice that there's three pumps, 1, 2, 3. Each pump, as the electron passes through, will pump 1 H+. So that means that if an NADH releases the electrons and becomes NAD+, it'll pump 3 hydrogens up across the membrane. But it only has 1 hydrogen itself. It's only got 1 hydrogen. So it's not like those hydrogens that are pumped aren't necessarily coming off of the NADH. And the same thing with FADH. Those hydrogens that are pumped aren't necessarily coming off of it. You know, if I jump back to the document camera for a second here, if you recall the setup here, it's these hydrogens that are flowing through ATP synthase that are going to come back and get pumped back up again. So the idea is to recycle these, which means that yes, these bring in new hydrogens constantly, but those aren't necessarily the hydrogens that are getting pumped up. You know, there's also this clearing that happens, clearing the hydrogens all the time out of the membrane using oxygen. So yeah, that's kind of an important concept in these pumps. Back to this, and here I am again. Okay, so that's the three pumps. They have names based on basically what they do. So this one, it takes the hydrogen away from NADH, so naturally it's called NADH dehydrogenase. Sometimes you'll see it called NADH oxidase because it's also oxidizing the NADH. Oxidation and dehydrogenation are a very similar concept in chemistry. So yeah, this is pulling hydrogens away from NADH. This one's called the cytochrome BC1 complex, and this one's called the cytochrome oxidase complex. Cytochrome is one of those interesting chemistry terms where back in the day they didn't have the skills, the tools to actually figure out what these things looked like, but they could tell that they were a different color than other ones. So cytochrome, cyto is the Latin for like cell, and chrome is the Latin for color, right? So so this is literally just cell color BC1, and this is cell color oxidase. So cytochrome C was the third, I guess, cytochrome molecule, the third colored thing that they found in the cell. And then this thing will oxidize it, so it's called cytochrome oxidase. And this one's BC1 because I guess there used to be something called cytochrome B. Maybe ubiquinone was called cytochrome B. And it would work on that and turn it into cytochrome C or something like that. Ubiquinone is also a funny name if you think about the word ubiquitous. What they found was this was just everywhere in every cell they tested, so they just called it ubiquinone because it was a ketone and it was everywhere in the cell. So these are the pumps and the shuttles. These are the 3 pumps. 1, 2, 3. Yeah, so the 3 pumps: NADH dehydrogenase, cytochrome BC1 complex. Other people call it stuff like ubiquinone oxidase, just to keep this going. Like some people call this NADH oxidase, ubiquinone oxidase, and cytochrome oxidase. That's maybe a better way to do it because it just tells you what it's oxidizing each time. This one's oxidizing NADH, this one's oxidizing ubiquinone, and this one's oxidizing cytochrome. Yeah, there's different ways to say what they are. But yeah, 3 pumps. Each one pumps a hydrogen. The other thing that's important here are the electron shuttles. So we're going to jump back to the front cam, pull up the learning goals, and now we're talking about the electron shuttles. The electron shuttles. 2 electron shuttles. Right, 2 electron shuttles. And the keep in mind is the shuttles are small compared to the pumps. The shuttles are very small compared to the pumps. And that's very important. So why is it important that the shuttles are small compared to the pumps? Well, if we jump back to this view, the pumps, the big pumps, have to sit in the membrane. They're across the membrane. They're these big chunky things. In order to pass the electrons from one to the other, they would have to like bump into each other, basically. And it's kind of awkward. So what nature has seen to do is get these little molecules here that literally just grab the electrons from here and push them over here. That molecule's entire job is just grabbing electrons from one place and bring them to another place. That's why it's called a shuttle, right? Think about like an Uber or something like that. It's like an electron Uber. It just grabs things and moves them. It doesn't pump anything. It doesn't have any kind of action like that because it's small, right? These guys are big because they have to have these channels for the protons to go up. They have to have a series of molecules to slowly kind of like bring up the hydrogen ions. We'll see that later. But these guys just have to grab grab electrons and push them over. So ubiquinone and cytochromes here, these small molecules that bring stuff over. And you know what? I think I should just find a structure of it. Ubiquinone structure. All right, here's ubiquinone. That's there's a good picture of it. This is ubiquinone. This repeats, so it's actually kind of a long chain here, but it's a lot smaller than something like a protein. So you can see that it's going to be very short. Ubiquinone also has a different name, which Wikipedia nicely has it as here, coenzyme Q10. You might have heard of this in different face creams. It's an antioxidant that's used in a lot of face creams because it's so good at grabbing electrons. So those electrons that it grabs can't go on and mess up your skin and stuff like that. And it does that. Okay, so that's ubiquinone. Let's talk about cytochrome C. Cytochrome C. Here's cytochrome C. Oh, let's go to Wikipedia for it. It's a small protein. It's a little protein, but that's still much smaller than than any of the protein pumps. So like NADH dehydrogenase, right? NADH dehydrogenase is absolutely massive. I can't find a picture. Oh, there we go. You see that picture of it. It's huge. It's very, very, very big compared to the sizes of other things. Okay, so that's the electron shuttles. The shuttles are small compared to the pumps. Now we're going to watch a video that puts this all together, and I'll break into the video every so often to explain what's going on. So now we're going to change our current topic to putting it all together. Putting it all together. And we're going to keep the same, keep in mind, we're going to do the same, the shuttles being small compared to the pumps. You can watch them carry out their electron shuttle function. So jumping up into this video. Obviously this video will have a link down below, down in the description, so that you can watch it on your own time if you want. So here's the video. I'm going to go just full desktop view and mute my microphone so that we can just watch the video, and I'll pop in every so often to explain. So I'm muting my microphone and we're going to watch this video.
All eukaryotic cells, from yeast to those that make up the human body, contain membrane-bound organelles with specialized functions. Mitochondria are double-membraned organelles that harness most of the energy that cells need to grow and reproduce. Nearly all of this energy comes from reactions that take place at the inner mitochondrial membrane. One of the key roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing a concentration gradient to be maintained where the intermembrane space has far more protons than the matrix. The membrane also contains a large protein complex called the F1-F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4.
Okay, I'm back off mute just to show you this whole thing. So down here is actually the outside of the mitochondria, and in here is the inside of the mitochondria. So we're kind of looking at this backwards. This is the ATP synthase where the H plus is rushing in, and these are the pumps as mentioned in the video. You can actually see the little green ATPs floating in to this guy, and as that turbine rotates, it kind of like kicks this thing into that little scissor or plier kind of conformation, and it jams the phosphates right onto the ATP. But that won't work unless there's a gradient. So that's what they were doing here. Here it's saying, now there's no gradient, now this thing can't go, which is why we have these pumps to constantly remake the gradient. So what you're seeing there is exactly the same thing that I diagrammed over here, right? Here's the ATP synthase, there's the pumps. And like I said, they're actually right next to each other. I'll put up the screen. Right, they're actually right next to each other, and they're all over the place. So it's not really like this. It's organized much more like the video is showing it. Okay, back to the video. And I'll take this away and I'll mute my microphone, and we'll continue watching the video together. So.
Proton pumping requires energy, and the 4 protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the 4 complexes are collectively referred to as the electron transport chain.
Okay, back off mute, and this is a really good kind of x-ray of the whole process. You can see the big proteins and the little tiny shuttles, right? The little tiny shuttles. So that's the point of the shuttles. As mentioned up here, the shuttles are small compared to the pumps. So these guys are pumping the protons, but these are important to pull the electrons along. They've actually got a nice flow of this. The electrons actually flow like that. Notice there's 2 pathways. One of these is for NADH and the other one is for FADH. NADH can activate all 3 pumps. FADH, being a lower energy molecule, as mentioned before, can only activate 2 of the pumps. So now they're going to track NADH and FADH as they go through this. So I'm going to change the keep in mind to NADH activates all 3 pumps. FADH only activates 2. FADH2 activates 2. So we'll save that. There we go. And you guys can watch that in the video now too. So I'm going to put this back to desktop. I'm going to mute my my camera, and here we go.
Focus on complex 1. A byproduct of sugar metabolism called NADH deposits 2 high-energy electrons in complex 1, where they are passed along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal 2 reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons.
Okay, back off mute. What you're seeing here is thermodynamics. If you remember, one of our curriculum expectations here is to talk about the laws of thermodynamics and how they apply to this process. As these electrons fall down to lower and lower and lower energy, so higher affinity molecules, that energy has to go somewhere. That's the first law of thermodynamics. Energy can't be created or destroyed. It has to go somewhere. And that gets stored up kind of as tension in the protein, as electrical kind of tension in the protein. And then when enough of it has been stored up, it shoots protons up into the intermembrane space where they can then do work. So that's how the laws of thermodynamics that we learned, stuff like bond energies, contribute to this process. So that's something that's important to talk about. Hey, back to the video. Sorry for all the interruptions.
The last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule. Complex 2 is similar to complex 1 in 2 important ways. First, high-energy electrons also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons. Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through 2 redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored each protein complex in isolation, but in reality they are very densely packed.
Okay, last interruption on this video. This is what it actually kind of looks like. There's the turbines, there's each pump, there's other turbines over here, all the shuttles are doing their work running around the membrane, passing electrons, and there's no specific chain for each protein. The ubiquinone and the cytochrome C will just pass the electrons that they're shuttling to whatever happens to be closest, and they're just moving randomly through the membrane. So it's not quite the same as the picture here. It's not the same as the picture where it's like clearly they're all in a row. It's not like that. It's very much everywhere. And that's that. We'll let the video finish here, and then I'll be back in a sec.
Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant.
Okay, that took longer than I thought to explain all that stuff. It is a pretty complex topic. So if we look back at our learning goals, we've talked about all of this stuff and we've put it all together, but I don't think I'll have time for uncoupling the electron transport chain today. We'll do that on Tuesday's video when we talk about some of the other types of metabolism that we can get into. Things like those bacteria that live where there's no oxygen and they can still manage to metabolize things. So really quickly, I'm going to jump over to the summary slide of this note. So takeaway learning goals and the summary slide, blah, blah, blah. Here's the summary slide. And what's important here is this keep in mind piece where NADH, notice it'll pass electrons through all three pumps. So 1, 2, 3. It'll pump 3 hydrogens. But FADH doesn't activate this pump. It actually passes its electrons directly to ubiquinone, to cytochrome Q or ubiquinone. And that will only activate these 2 pumps, which means that NADH pumps 3 protons, FADH pumps 2, meaning that one NADH can make 3 ATP and one FADH can make 2 ATP. Another thing to note is that it takes energy to bring the NADH from glycolysis in, so you kind of lose 2 ATP bringing in an NADH. Lose 1 ATP bringing in an NADH, so that actually reduces the amount of useful energy that you can get out. So I think there's somewhere in here, here's the overview, right? Each NADH can make 3 ATPs. That's why these 6 NADHs can make 18 ATP, because every pump is 1 proton and every proton is 1 ATP. Each FADH only makes 2, so 2 FADHs make 4 ATP. And then glycolysis, which happens outside the mitochondria, remember that happens in the cytoplasm, those NADHs have to get into the mitochondria, so they lose a little bit of energy on the way. So those NADHs are special. They can only make 2 ATP each, basically. So your maximum is 36 ATP. Really, your maximum possible is 38 if these NADHs stay intact on the way through. So sometimes you'll see 38 as the maximum, sometimes you'll see 36 as the maximum. But really the process has a little bit of loss in it. Sometimes protons will leak back through the membrane, sometimes things just go a little bit wrong. So really you're looking at like 30 to 32 ATP per glucose. This is per glucose. And that's where we're going to end. So just as a quick review, we did the importance of the proton pumps to maintain the gradient, and that was based on this this diagram I had, right? They have to cycle the hydrogens through, the protons through. We talked about NADH and FADH, those molecules. So we talked about the molecules, NADH and FADH. We talked about the 3 proton pumps, NADH dehydrogenase, cytochrome BC1, and cytochrome C oxidase. And we talked about the 2 electron shuttles, which are ubiquinone and cytochrome C. Really what's important there is why we have those electron shuttles, the idea that they're small molecules and that they pass electrons along. And we didn't get to uncoupling the electron transport chain. That's going to be on Tuesday's video. So yeah, thank you very much. I'm going to go write the quiz for this video. Please do it so that you can get the time for today's lesson. Thank you very much, and I hope you enjoyed it.